Dental and Craniofacial Research at NIH, where he continues his work on several aspects of calcium signaling and epithelial transport mechanisms in health and disease. Samuel has generated very stimulating and inspiring papers about the involvement of TRPC channels in calcium influx, the interaction with steam and ORI, and the role in cell function. The second speaker, Hyunjin Kim, finished her PhD in the Department of Physiology at Seoul National University College of Medicine in South Korea, and then she moved to the lab of Samuel at the University of Texas. In 2010, she moved back to the Sungkyunkwan University School of Medicine, where she is an associate professor. Today, she's going to talk about the regulation of autophagy by the PI3 phosphate TRPML3 interaction. But before that, Samuel is going to talk about his most recent publication concerning TRPC3 channel gating by lipids requires localization at ER plasma membrane junctions defined by STIM1. So, Samuel, the floor is yours.
It says that I am not. Let me try it again. Okay, I'm assuming you can see my screen. Thanks everybody for attending, and therefore thanks for the invitation to tell you about our work. It's just a short note that these papers, this work just came out in JCB, so if I'm missing something, should be available now. So our interest mainly is what happened to ion channels transporters in junctions, and mainly this started from looking at secretory cells or polarized cells. In this case is the pancreatic acinar cells. Let me see if I can get pointer. The pancreatic acinar cells, which you can see, it's a highly polarized cells with the apical membrane, the basolateral membrane, and many of these signaling proteins like the ORI, the IP3 receptor, STIM, all of them are expressed in this apical region, apical junctions, and these include the TRPS channels. So it is obvious that localization in those junctions are very important, first of all, for the functions of the cells. This is the area where exocytosis takes place, and obviously you want to have a lot of activity there, of calcium activity for the secretions of these cells, or cyclic MP, which is cyclic MP-mediated secretions. And the question is how you organize these things into this membrane contact site, how these localization membrane contact sites affect these channels or transporters and the function and the cell functions, and how lipids within these junctions are affecting cell function and the channel function. So it's just a brief things about those ER plasma membrane junctions. It's where the ER come close to the plasma membrane. These junctions are held by tether proteins that I will talk a little bit about the or extended synaptotagmins. I will say one word about this anoctamin 8, which also functioned as a tether within these junctions, and a note about STIM, and show you that STIM actually it's a very important tether in the function in the ER plasma membrane junctions, and how it can affect channels that either directly or indirectly. So this is an example of the e-synaptotagmin functions in the junctions. This is an example where one of the proteins that control the function of STIM1, the SARAF protein, for it to interact with STIM1, it will not interact unless it is in the junctions. It is STIM or I-form junction. And when we expressed E synaptotagmin 1, you can see that it becomes constitutively localized in the junctions, and when you knock out E synaptotagmin 1, it cannot make it to the junctions. This has functional consequences. These channels tend to inactivate, as all of you know. The OI channels are has two types of calcium-dependent inactivation. This is the slow inactivation that becomes very fast when you co-express E synaptotagmin 1. The other things that is shown here is very specific for E synaptotagmin 1, because when the EC2 and EC3 don't do it in this case. The second protein is the anoctamin, and this is shown another way to show the functions of anoctamin 8 within the junctions. When we knock it down, you see that there is a very marked reduction in STIM OI complexes and a reduction in the current. And when we co-express it, overexpressed in the cells, there is a dramatic increase in the current, and the current becomes very quickly inactivated. Even when we have 10 millimolar bapta in the pipette, and most of this inactivation, it's because also SARCA pump get recruited to the junctions to close up this activity. So we, as you know, that we have been studying for quite some time the role of STIM in the regulations of TRP channels. And in some work in 2014, we found the two mutations at that time. We didn't know really where they are localized that well. Two mutations that have profound effect on the interactions of STIM and TRP channels. One of them is this in the 241, 245 region. There is a coil region here, which is very close to the TRP domain, that dramatically enhance the interactions of STIM and OI, while a mutation in what is now known is the vertical helix or the pole helix that is found actually in every TRP channel. That mutation in this junction completely eliminate interactions of TRPC3 with STIM. And not only that, even when you have this enhanced interaction, even this one can be destroyed. More recently, we start looking what happened to response or to the function of STIM or the interaction between STIM and OI when we look at lower or different type of agonist concentration. And you see here that when you express STIM1 with TRPC channels, you can get a dramatic increase in the activity. Same thing is when you use this particular mutant, while this mutant completely lost the ability to respond to STIM. When Hai-Ping joined the lab, she looked at the project and she wanted to actually study the possibility that there must be something about STIM and about interaction between STIM and TRP channel, which is not a standard interaction, just changing the activity. So she did a dose response curve to using these three proteins, one which interacts, it's a normal interaction with STIM, enhanced interaction, and no interaction. And as you see here, she found something quite dramatic, that there is no effect, almost no effect on the Vmax, but there is a dramatic effect on the apparent affinity for muscarinic stimulation of this channel, whether when this mutant has very much reduced affinity and this mutant has much enhanced affinity. This turns to be mainly because of enhanced interaction between them and not enhanced surface expression. There is some increased surface expressions of this particular mutant, but it's actually very mild, very small interactions. On the other hand, these TRPC and STIM1, when you use FRET, you can see that there is very good FRET interaction, that it is enhanced by carbachol, but this mutant shows a much higher interaction with STIM1, and although there is carbachol stimulation cause slight enhancement, it's still very high FRET already. While this particular mutant has a reduced interactions with STIM1, actually reduced FRET in the basal level, and receptor stimulation doesn't really change it much. the last things that we did is, what she did is to look at what happened if you actually directly activate the channel with a ligand, which is similar to deacetylglycerol, and ask what happened under these conditions. And what she sees, there is an increase, a clear increase in Vmax, and even higher than what you see with the agonist. But there is actually very little effect, if any, on the apparent affinity of the channel for its own ligand. So most of the effect is actually, when you look at the substrate, is for activation of the channel. The question is, what happened now if you use a knockdown of steam? We got these cells for more. And what you can see here, that the dose response curve for all of the channels completely collapsed to this one, which is completely insensitive to steam. There is no effect of either the Vmax or on the apparent Km for receptor stimulation. We can completely rescue this activity, and you see it here, where you look again. This is another experiment which we look at the dependence of TRPC3 in steam knockout cells. And when you overexpress STIM1 in these cells, you see here you get a dramatic effect, almost no effect on the Vmax to speak of, but dramatic effect on the apparent affinity, a dramatic reduction on the apparent affinity in stimulations to carbon. Another really important finding in this, which actually came as part of the review of this paper, where we look at the reversal potential, and it turns that when you have steam there to take the channel to the junction, its reversal potential change. It becomes more selective, in this case for sodium over cesium, which is cesium in the pipette, sodium in the medium. And this is the case for either the TRPC3 or this particular mutant, the 241 mutant. But this particular mutant is not affected, and the reversal potential for all of them collapse to this one. So being in the junctions also determine the selectivity of the channel. And what gets it to the junction and what determines the presence in the junction is steam itself. Question is, do you need other tether in the junctions? And yes, they are. I just told you about ANO8, which is one of the tether in the ER plasma membrane junctions. When you co-express it and you stimulate with low receptor stimulations, you can get a very profound effect. And you can see it with TRPC3, you can see it with this particular mutant. And again, this mutant is completely resistant. And this is the SI, which is the reverse of this experiment. The next one is to look at the E-synaptotagmin, and the E-synaptotagmin is the same story as you see with the ANO8. There is a response and an increase of TRPC3, the L241S, and I807 is completely insensitive to changes in E-synaptotagmin, while again the SI E-synaptotagmin will have the exact reverse effect. Another important thing is, once we treat them with SI synaptotagmin, the current density of all of them become, you can see it here, become very similar, and this is the same thing for ANO8. So this tethers, or the junction has to be completely intact in order to see this effect of the mutant or the different behavior. Again, as I showed you with the STIM and OI, in this case as well, E-synaptotagmin 2, E-synaptotagmin 3 absolutely have no effect. This is not to say that they have no effect. We look at other channels, and there are other channels which are profoundly affected by any of these other E-synaptotagmins, but not these particular calcium-mediated channels or calcium-transporting channels. To look at what E-synaptotagmin is doing, again, we look at surface expression, and it doesn't seem to have much on the surface expression of the protein, but it really enhanced the interactions of STIM with TRPC3, that you can see some of it in the basal conditions that is enhanced by carvacrol stimulation. That's importantly, when you overexpress E-synaptotagmin, you see an enhancement without much an effect on the surface. And when you stimulate with carvacrol, you can get the maximum threat between them, indicating to us that what the junction is doing is allowing these channels or these proteins to come together. Another truly, for us, really interesting was this experiment. This is why we said that steam is the one that determines the junctions, and steam is critical in the junction. When we take steam knockout cells and we look at the effect of isonoctagmin 1, is completely eliminated. So the functions of all of these tethers require steam to form the junctions. Only when steam forms the junctions, or there are junctions formed by steam, you can get the other tethers, you can get the whole things assembled together. The next things we wanted to see what happens with lipid. Actually, we started with these particular experiments. The question was, we knew that PIP2, PI4, PIP2 is important to be in the junction, to assemble the junctions. When we deplete it from cells, we see that there is no ability, I mean, steam and OI will still come together, but you do not get similar type of regulations of the proteins when you deplete PIP2. So we decided to look at what happened when we deplete PIP2 from all of these constructs, and what you see it here, that depletion of PIP2 reduced the activity of all of these proteins to be similar to this one of the I807S mutant that is steam resistant. All the activity is the same, and again, the reversal potential. So being in the junction, stabilized by PIP2, determine the selectivity of the channels, that when we actually deplete PIP2, all of them collapse to this, to the same reversal potential. The next question was, what happened with carbachol stimulations? What PIP2 is required for when we stimulate with carbachol? And you see that carbachol stimulation now has two effects. With PIP2, so a depletion has two effects on these stimulations. One, it reduces the Vmax, and second, it increased the Kn. It's like having both effect of these two proteins. The increased Km and the reduced Vmax, both of them happens when we deplete from PIP2. Another interesting experiment for us was that when we deplete with PIP2 and we look at the reversal potential for direct stimulation of the channel, we didn't see much of an effect. This actually turns to be slightly statistically significant, but most of it, the direct activation of the channel did not care, I mean, the reversal potential did not care about PIP2. Although when we look at the dependence of GSK stimulation, what you see here again, there is no effect on the Vmax, but PIP2 or present in the junction was required for access of the substrate to the channel to activate the channel. So we started asking a little bit more about what type of lipid, or how the lipids actually affect the channel. Do they only affect it directly by binding to it, or do they have an additional effect beside that? The first one is, of course, the supply of deacylglycerol, and whether there is some additional effect, sites on the channels. And about that time came out this paper, which is the first structure that found lipids within the channel itself. And they observed two lipids that showed up in the channel, one in the pore, what they call it site 2, and one next to this elbow that is also embedded within the lipid domain. And this particular site here, which the peripheral site, and they call it a site 1. In a previous work, before the structure actually was available, Klaus Groschner, already based on just modeling after the first trip channel or trip V channels was actually published, he identified this particular residue as being important. It comes from an adjacent subunit, that this residue regulate interactions of the lipid with the channel. When these structures came out, all these 4 residues touch the lipid. For some reason, in case of TRPC3, mutation of this residue has actually no effect on the channel, although in all of the other TRPC4, TRPC5, TRPC6, in all of these cases, this residue turns to be very important. Maybe this particular domain, the 6 transmembrane domain, doesn't sit close enough to the lipid domain in TRPC3. However, those 2 that do attach the lipid, when you mutate them, you lose the effect, indicating that at least one effect or one role of PIP2 is supplying deacylglycerol to this site. The question is what happens in this particular site? And in this case, we did first mutations asking for, or looking, we mutated many more of these sites in this region, but we look only on proteins, on mutations that affect channel activity. All of these mutants, except this W334 residues, did not affect surface expressions, while this one affects surface expressions of TRPC3. Yet, this is the one that gave us the most dramatic activation of the channel. This particular mutation completely inhibited the channel, while these mutations have selective effects. This particular one just activates both the inward and the outward current, although the inward current was activated slightly more than the outward current. These particular mutations had absolutely no effect on the outward current, but affected the inward current, while these particular mutations affected the outward current with no effect on the inward current, which suggested to us that lipids in this region affect the shape of the pore to regulate the gates. Obviously, we don't know exactly how it does it, but it just indicated that lipids here may be important for regulations of the gates of the channel to determine the conductance. I'm saying that because when we look at the reversal potential with all of these particular mutations, we don't see any effect on the reversal, so the selectivity of the pore doesn't seem to change, only the open probability depending on which side of the channel will be affected. The next one was to see what these proteins will do to the stimulations by direct agonist and by carbol. And what you see it here, that these particular mutants, when we look at the outline on the inward current, we see a dramatic effect on the Vmax. Same thing when we look at the outward current. Look at the inward current, we don't see much of an effect on the Km, although in the outward current, we see a small effect on the Km. It's not as dramatic as you will see later, but it is a little bit, again, saying something about this region affecting the channel in a selective way, the different gates. When we look at the direct activation by GSK, and what you see here again, there is a strong effect on the Vmax on these particular mutations without affecting access of the ligand to the pore. Again, also here we try to see what happens when you remove PIP2. The first thing that we notice is that this particular mutation was still responding to PIP2. So it did not become completely PIP2 independent, but the effect on the Vmax, the effect of PIP2 depletion on the Vmax is completely disappeared. Actually, we might have got even slightly higher Vmax stimulation, but elimination of PIP2 now dramatically increase the Km, as if PIP2 is required for this mutation to control access of substrate to the pore. When we look at GSK activations, you see there are two things here. One, when you look at the depletion of PIP2, again, when you look at the Vmax in both cases, there is no effect on the Vmax, but even this dramatic effect on the lipids was completely restored the affinity for GSK was completely restored when we did these particular mutations, namely allowing access of GSK, or this particular mutant allowing better access of GSK to the pore. However, there was a problem in when you stimulate these cells with carbachol, when carbachol stimulation did not allow a proper access of the channel of the either of the receptor, the substrate to the pore, or proper activations of the receptor. We needed to see, or we thought at that time that maybe this particular mutation, because it gives us such an activation, it is the mutation that determines localizations of the channel in the junctions, because TRPC3 need to get to the junctions. The question is how it gets there. By looking at the effect on the behavior of the channel in the STIM knockout cells, you can see here that these two particular mutants still can increase the inward current independent of the presence or absence of STIM. We can still see an effect on the channels on it increased Vmax with slight reductions in Km in STIM knockout cells. And when we look at eSynaptotagmin and SI-ANO8, when we knock down eSynaptotagmin or ANO8, we can see that they still have an effect, namely that these particular mutations for us telling us that it is not regulating the localizations of the channel in the junction. So we look for other possible sites, and we identified in one of these linker helices, a linker between the helices, the cytoplasmic helices that are actually surround this pole helix, that it's called the FFAT site, that can accept either phenylalanine or the Y, a Y can replace the F in this particular site, and the FFAT site stands for 2-phenylalanine and an acidic domain, and this is the acidic domain within these sites. So we decided to see if this particular domain is important. Now, this domain is known to interact with a VAP protein that sits in the endoplasmic reticulum and interact with the protein and recruit them into the junction. So we asked whether these FFAT sites have the same function in the TRPC3. And what you see here is, first of all, that TRPC3 does interact with VAP-B. Actually, it doesn't interact much with VAP-A, as specificity to VAP-B. And when we mutate this phenylalanine, the FY sites, we lost completely the interactions. And importantly, the interaction with VAP and another one of these tether in the ER plasma membrane junction was completely dependent on STIM, one forming the junctions, because when you look at STIM1 knockout cells, you lose almost completely the interaction. And you see here, as you would expect, when you add VAP-B, you see here an increase in the current. When you knock out the VAP-B, you see a decrease in the current, as you would expect them from being or getting outside the junction. If they get outside the junctions, one would expect them to change the affinity for carbachol stimulation. So we look if this can be the case, and to do that, first we mutated these two residues, and we looked whether they lost the ability to respond to VAP-B, and what you see here, this is the case. This is the TRPC3, this is the response to VAP-B, and when we look at this particular mutant, we no longer see any effect of VAP-B. When we look at what happens to carbachol stimulation, you see here there is a small effect on the Vmax. This protein doesn't really express that well in the plasma membrane, but the most dramatic effect was an increase in the Km for carbachol stimulation, again indicating that the channel has to be in the junctions to be properly stimulated by carbon or by any kind of receptor stimulation. So let me lead to summary my presentation, indicating that this channel, TRPC channel, I also want to indicate that some of these sites that I was showing you, all of these residues that determine functions of this lipid and these lipids on these FFAT sites are completely conserved in TRPC 3, 6, and 7, and partially conserved in all of the other TRPs. That lipids binding to this site, that is the pore, lipid binding to the pore site, is PIP2 provide lipids for activation of the channel by diacetyl glycerol with this site. When this particular lipid has at least two roles, one of them is to control access of diacetyl glycerol to this particular site and to stabilize the TRP channel, to allow the TRP channel to stabilize and stay in the junctions. And this FFAT site is the one that interact with VAP-B in order to stabilize or keep TRPC3 in the junction. I need to just thank these guys for all the work that they did. It takes them for quite some time to finish this work, and thanks more for providing us with the STEAM1 cells, STEAM1 knockout cells, and I will be happy to later answer questions. Obviously, the funding comes from the NIH. I will stop sharing now so that Hyun Jin can go ahead with her presentation.
Okay, I'd like to thank ECS webinar organizers and Dr. Shmuel Muallem for giving me this great opportunity. And also I'd like to thank Professor Juan Rosado for nice introduction. Today I'm going to talk about regulation of autophagy by the PI3P3-ML3 interaction. So, okay, let me give you a brief introduction about autophagy. And in step 1, mTOR inhibition by cellular stresses such as starvation, it can initiate autophagy by sequential activation of ALK1 complex and class 3 PI3 kinase complex. As a result, PI3P is accumulated in the isolation membrane or phagophores, forming autophagosomes. In step 2, PI3P recruits its downstream effector proteins with many ATG proteins for phagophore expansion and sealing. At this time, phagophore is elongated by fusion with multiple membrane sources such as mitochondria, recycling endosome, and Golgi complex. After sealing, phagophore is called autophagosomes. At this moment, ATG proteins, most ATG proteins leave autophagosomal membrane, except ATG8, also known as LC3. So we can use ATG proteins as phagophore markers and LC3 as a whole autophagy marker. In step 3, autophagosomes mature by sequential fusion with endolysosomes to be autolysosomes. So as you see here, autophagy is full of fusion events from autophagosome formation to autophagosome lysosome fusion. Now you are seeing the figures showing membrane fusion machineries in autophagy. Autophagy membrane fusion events also follows general principle of vascular transport. So many coat proteins, RepGTPase tethers, snails, and phosphoinositides were extensively studied in autophagy. Although it is well known that membrane fusion in autophagy is calcium dependent, how calcium is provided for this process and the source of calcium are not known yet. So in this kind of intracellular membrane fusion reactions, a focally localized and transient calcium signal is required, not global calcium increase. Therefore, the calcium release channel should be closely placed to the fusion sites. We thought our TRPML3 is the best candidate for the calcium release channel. Actually, I started this study on TRPML3 almost 15 years ago when I was Dr. Muallem's postdoc. We together found that TRPML3 is expressed in the plasma membrane and endocytosis and autophagy pathways. And that TRPML3 is a calcium permeable cation channel that is inhibited by acidic pH. We also found that activation of TRPML3 increases autophagy, but its inhibition suppresses autophagy. So at present, TRPML3 is the only ion channel that has been reported to colocalize with autophagosomal markers. After I became independent, I found that TRPML3 interacts with mammalian ATJ homologate 16 to increase autophagogen formation. As shown here, KT16 with the LC3 is involved in PHAGO4 elongation and sealing. I also found that TRPML3 is recruited to autophagogen formation sites by pymetylation. All these findings suggest that TRPML3 may be the calcium release channel that provides calcium for the fusion process in autophagy. To test this hypothesis, I planned this experiment. We wanted to see how starvation regulates TRPML3. As shown here, only TRPML3 expressing cells responded to acute starvation, even in the absence of extracellular calcium, suggesting that this calcium is from intracellular compartments. And this calcium increase was not affected by TPC antagonist, but completely abolished by TRPML antagonist MLSI1. So we could think that TRPML3 is really involved in this acute starvation-induced calcium increase. To better study the role of TRPML3 in autophagy, we generated this fusion construct. As you can see here, you can see this nice increase of TRPML3 activity by agonist stimulation. Then we tested its specificity by this calcium experiment and found that GCAMP6 fused with TRPML3 does not respond to global calcium increase induced by CPA or GPN treatment. So now we have a specific reporter of TRPML3 mediated calcium signal. So we tested if TRPML3, where TRPML3 is active during autophagy. As shown here, TRPML3 was not active in the mature autophagosomes, but almost fully active in the PHAGO4. We also found that starvation increased TRPML3 activity here. So this result suggests that TRPML3 can be activated by a autophagy signaling molecule. We suspected PI3P because PI3P is accumulated in the PHAGO4, where TRPML3 is active. First, we tried to see how quickly PI3P is accumulated upon starvation, and found that this PI3P level is very good in agreement with the TRPML3 activity. So next, we investigated the relationship between TRPML3 and PI3P and found that N-terminus and loom region of TRPMS3 binds to not only to PI3P but all 7 phosphorinositides. This was rather disappointing because these PIP are very expensive. Anyway, we had to buy all 7 phosphorinositides to deliver them into the cell, expressing TRPMS3, Camp6, and LC3, and found that only PI3P significantly increased the GCAMP6 signal and LC3 particles at the same time. This result suggests that not only PI3P not only interacts with the TRPMS3, but PI3P can activate TRPMS3. To test if TRPMS3 directly activates TRPMS3, we performed wholesale patch clamp experiments. And as shown here, extracytogenic PI3P application does not activate TRPMS3 that much. However, cytogenic PI3P increased TRPMS3 basal activity, like here. And only in this case, extracytogenic PI3P application induced huge TRPMS3 current. We also did inside-out macro patches to see if cytogenic PI3P directly activates TRPMS3, and found that immediately after PI3P application, TRPMS3 is activated almost half of the maximum current. So from these results, we thought TRPMS3 may be the downstream effector of PI3P to regulate autophagy. To test the possibility, we inhibited TRPMS3 and added excess PI3P into the cells and monitored autophagy. As shown here, PI3P effect on autophagy was almost completely abolished by TRPMS3 inhibition by dominant negative construct or siRNA. So we thought TRPMS3 is required for the effect of PI3P on autophagy. Next, we inhibited upstream and activated TRPMS3 and monitored autophagy, and found that autophagy inhibition by class 3 PI3 kinase inhibitors was rescued by TRPMS3 activation. So from these results, we thought TRPMS3 is really downstream effector of PI3P. If TRPMS3 is a downstream effector of PI3P, it must have its binding sites. We found that PX domain in the N-terminus of TRPMS3 and RXL motif in the loom region. And charge removal mutations not only inhibited PI3P TRPMS3 interaction, but also suppressed autophagy. And even in this case, agonist stimulation of TRPMS3 rescued almost completely the autophagy. So we conclude that PI3P TRPMS3 interaction is very important for autophagy progress. Here is a summary. Upon induction of autophagy, PI3P is accumulated in the phagophore membranes. PI3P then recruits and activates TRPMS3 to release calcium from the phagophore lumen. So we think this calcium release via TRPMS3 facilitates autophagogen formation by triggering membrane fission events. So I'd like to finish my talk by saying thank you to my lab members and collaborators. Most experiments were done by Soon here. She earned a PhD degree last year. And other lab members also contributed to this project. And Dr. 서병창, 서인석, and of course, Dr. Shmuel Muallem gave me a lot of help. Thank you for your attention. I will stop sharing my slides now.
Thank you very much, Samuel and Hyun Jin, for your really interesting talks. Now the talk is open for questions, so you may raise your hand if you want to make any question. We have a question from Sandy Patel. Sandy, when you come.
Actually, I think Barbara was first. Barbara, do you want to go first? No. Okay.
Go ahead. Go ahead.
Okay. I have two questions. One for Shmuel. Shmuel, really interesting stuff. I have a question about the ion selectivity changes. So most of the measurements that you made, I'm not sure how you were recording, but you mentioned sodium and cesium. But what about calcium? What's it doing to the calcium selectivity, and how relevant is that for its signaling function?
One of the mutant, actually, when you don't have steam, you increase the calcium selectivity of the channel. When it is outside the junction, it becomes a much more calcium selective. The other way to do it is to look, actually, it turns that this is studies that are ongoing at this time, that the pole helix, there is a hydrophobic region within the pole helix that is very important in getting calcium-dependent inhibition of the channel and calcium selectivity. So within that region, maybe something happened to it when it is outside the junction, and then you can get calcium permeability of the channel seems to increase. In a way, for us, this is a disease type of behavior when the channel actually get out of the junctions, which is normally you would equate with some pathological condition. There is too much calcium that can come in and obviously induce cell toxicity.
Right. So the calcium permeability is changing. Okay.
Calcium permeability is changing when it is outside the junction. Yes.
Yeah. Okay. Yeah. Interesting. And I have another question about the autophagy stuff. So
Yes.
where is the calcium in the phagophore, and how does it get in?
Actually, phagophore is connected to ER lumen, and multiple vesicular stuff from organelles will have some calcium inside. So I think the calcium is enough to initiate membrane fusion.
So the calcium still replete in the forming phagophore. Has that ever been measured? I don't know the literature.
As far as I know, nobody measured the inside the phagophore calcium.
That's interesting. Thanks very much.
Okay, thank you. Barbara, you're next.
Yeah, thanks both of you, and very interesting talks. First question I have for Shmuel is, so if STIM is so important for tethering, a lot of the lipid transfer takes part at the ERPM junctions. Wouldn't you expect a loss of STIM to then maybe lead to depletion of PIP2 or a reduced production of DAG? So how much of these effects are really due to a direct interaction between TRPC3 and STIM1, or perhaps due to the defective tethering and defective transfer of lipid precursors and a reduced production of DAG?
Well, again, it depends on how much of PIP2 you are depleting. Obviously, if you deplete too much PIP2, you will not have the same type of activation. However, we did not expect to see, we expected to see a reduction, what you would call in the Vmax or in the maximum activity, if we don't have enough substrate. We did not expect to see a change in the affinity for the substrate. And because we are seeing dramatic change in the affinity for the substrate when we activate directly by GSK or deacylglycerol, we conclude that actually the channel, that the pore is changing also. It is not just because of simple depletion of TYP2. We are also not depleting it to the extent that there will not be substrate, because then you will not get enough activation no matter what.
Yeah, it might be interesting. I think there's now a new DAG sensor out, is to really look at levels of DAG instead of PIP2 as well, especially for TRPCs. Great. And a quick question for the for Hyun, the PI3P. Which cells were you looking at? So the levels of PIP3 change depending on the cell type. So neuron has a lot more than a HEC cell. Which cells were you doing your experiment?
We used the HeLa cells and 293 cells for the experiments, and I'm not sure about neuron.
I just thought there's a very recent paper by Volker Hauke saying that neurons have really high PI3P levels compared to other cells. So could be interesting. I'm not quite sure about the role of TRPML in neurons. Okay, thanks.
Thank you.
Okay, thank you, Barbara. Christian Grimm.
Yeah, first of all, thank you very much to both speakers for great talks. It was really interesting. I would have a question for the second speaker on the PI3P data on TRPML3. So we have already published in 2017 that PI3P does not activate ML3 in endolysosomal patch clamp experiments. I can send you the link to the paper if you're interested. I was just wondering, how can we figure out the differences here, obviously, in settings or cells used, or what were the conditions? I assume you did not do endolysosomal patch clamp, but plasma membrane redirected patch clamp?
I've never tried endolysosomal patch. I think a low pH inside the endosome may inhibit TRPML3 activity.
But we measured under ideal conditions. That means increased pH, so higher pH, because as you say, low pH is blocking ML3, and also high potassium, because then the currents are much larger. And we only saw activation with PI3,5P2 at the time. Yeah.
Okay. I'm not sure about the endosome stuff, but I used the whole cell patch clamp techniques, so maybe that is different from your condition.
Yes, but naturally you would expect the channel to be in endolysosomes, in early endosomes and in late endosomes and lysosomes, not in the plasma membrane.
Sure. Yeah.
So something to
But I also think that Hyun Jin showed that you have to stimulate the cells first in order to see the PI3 activation of the channel.
Maybe I missed this point. Okay.
Yeah, it's not an artificial system. The way she does the experiment is a little bit different from what you guys were doing.
Okay. Is this going to be published soon, or then we could have a closer look at it and try to see what?
This is in revision at PNAS.
Okay, good. Thank you.
Okay. There are no more hands up at the moment, but we have a comment and a question by Rajesh Bhargwah, through the chat. Thank you for the nice talk, Samuel. It was a lot of data, and I might have missed it, but I wonder if you have assessed whether there is a change in the expression level of muscarinic receptor 3 in cells or the TRPC3 mutants, and second, if the mutant W334A also shows a shift in the reversal potential under GSK tricky conditions.
So the answer is, for the first one, yes, it is in the paper, and the muscarinic receptor does not change at all. And I mean the surface or the total, and the 334 mutant does not affect the reversal potential by itself. Either, no matter how you activate it, it doesn't have any effect on the reversal potential. So, I mean, this particular mutation seems to increase channel open probability, although we didn't measure single channel, but we do not see effect on access to the access of the substrate, or even not surface. On the contrary, surface expression of this channel is dramatically reduced. So even the current that we see is quite downregulated. I mean, underestimated.
Mhm. No more questions at the moment. Samuel, the data you showed are made in pancreatic acinar cells?
Ask me again. Sorry, I didn't get the question.
The cells that you use in your experiments
Yeah,
are
no,
pancreatic acinar cells?
no, those are all. These are all HEK cells and transfection in AK cells, HEK cells, pancreatic acinar cells. The TRPC current is really tiny. I mean, the protein is expressed actually quite well, but the current is very difficult to record.
Okay. I don't remember very well from your first slides when you showed the localization of STIM1, TRPC3, I remember, and
Yeah.
probably RAI1, right? Where was located TRPC3 in the pancreatic acinar cells, in the apical membrane or in the bilateral membrane?
Well, we thought in the beginning that it is on, we claim that it is probably in the apical pole, on just in the lateral membrane. But now I actually think that it is in the apical membrane itself, because in the duct there is no ambiguity of where it is, and it looks completely apical. So I think it is apical. Also, one of Indu's former students or postdocs look at TRPC3 expression in MDCK cells that are born in a polarized manner, and it's only in the apical membrane. So I think it's actually TRPC3, OI. There is a lot of them in the apical membrane.
Okay, that's really interesting. Apparently, there are no more questions or comments through the chat. So if you don't have more comments, we can end the talk. Oh, there is one more question by Barbara.
Yeah, just a quick question, Shmuel. Have you ever tried looking at the TRPC regulation in the ORI deficient HEK cells?
No, we did not. We have them now, but we did not look at it, which will be actually obviously interesting to see if you need to actually make the junctions together in order.
Right.
But it's actually, for me, it's just, again, I'm throwing something even that was not asked. This business of steam regulating the junctions, almost everything we looked at that works in the junction does not do it without steam. No matter how much other tether you put, no matter what you do, if you don't have steam, there is no regulation. There is nothing goes to the junction without it.
Okay, so if there are no more questions, thank you all very much for attending the webinar. Thanks, Samuel and Hyun Jin, for the really, really interesting talks. So I think that we can now finish this webinar.